I work on brain on a chip, and how we make the brain is we have a couple different systems. One is called the central nervous system. We can divide them up into different sections. So example, we could have the hippocampal brain cells along with the cortex cells in different areas. The other chip that we have that's part of our brain on a chip is called the blood-brain barrier. And what the blood-brain barrier is, it is essentially the blood vessels in your brain. And what they do is they sort of serve as bouncers, meaning they only let certain things go to your brain, which is a good thing. But what makes it challenging for people that are developing medicines is that the vasculature in your brain doesn't always let all types of medicine go through. And so that could be at a detriment, because sometimes you want to help somebody who may have a brain tumor, and you have to be able to figure out how do you, get past the bouncer, how do you get past the blood-brain barrier to be able to treat the person. So what that chip looks like is we have a hollow fiber that we see endothelial cells, and so those are the cells that make up the vasculature. And then behind them are the astrocyte cells, which are the support cells, meaning they're the ones that help the endothelial cells out. And so we put all of these on a fiber that we then connect to fluidics. Because biology is very dynamic. There is fluid moving through us all over the place. Even in between our tissues, we have what's called interstitial fluid, which is very, very slow movement of fluid. And so we need to make sure that when we're culturing these cells, when we're making or printing whatever these tissues are, that we don't just stick them in a petri dish, because that's old school biology. People are very dynamic. And so you need to make sure that you are giving the cells the same kind of environment outside the body. So a lot of what I do is I work on the fluidic side of it as well, connect up the chip, make sure that we perfuse it, meaning that we introduce flow, because the cells are very sensitive to that. They know that as soon as they have flow, that means that they're going to do something. So in the case of cells, for example, they will line up in the direction of the flow for the endothelial cells. So that's what the chip is. It's a hollow fiber with cells grown on the inside, also on the outside. And on the outside, we can also put the neuronal cells. So now we have the endothelial cells, which are the vasculature cells. And then on the outside, we have the brain cells that are the neurons. And so we can then introduce the different drugs and see, do those bouncers or that blood-brain barrier, is it going to allow it? And then what's going to happen on the neuronal side? And we can see what happens on the neural side using these probes that we can help record the signals of how these cells ended up talking to each other, or what were the chemicals that they secreted, and can we measure those levels? And so that's what our probes do.